Can anyone see slides?
Yeah, now
No?
we got slides. Yeah.
Everyone can see
Okay.
that, yes.
Yeah.
I'm sorry.
So
You
you
guys
thought
should
you
have.
were in the radio. We are not in the radio. That's
Okay.
we have video. Okay, but
I was sure this you were seeing. Okay, so I just go through faster now. So let's begin. And as I was talking, animals are exposed to seasonal lack of resource, and metabolic depression is a widespread strategy to survive during these periods of lack of environmental resource. And we have that several examples from hibernation to estivation, freezing, hypoxia, and air exposure of aquatic organisms. So animals suppress their metabolism by decreasing the rate of ATP expensive processes. So the idea is to spare resource and prioritize only critical pathways or processes for survival. And in this scenario of global suppression of transcription, translation, and active transport, some genes are selectively activated. And as I said, without slides, we have that microRNAs and epigenetic modifications are some upregulated pathways that help to suppress the, the energy expensive processes. And on the other hand, we have the upregulation of transcription factors that drive the expression of chaperones, antioxidants, and some other pathways involved in the cellular stress response. So in 93, work with garter snakes. Here, this is in Portuguese, I apologize, but this means freezing, and this means anoxia. This was the first observation of the upregulation of antioxidants during metabolic depression. And a few years later, this was observed also in other species and other conditions of metabolic depression, freezing, anoxia, estivation. And this phenomenon of increased antioxidants during metabolic depression was coined as preparation for oxidative stress. And since the proposal of this hypothesis, there was it's not a belief, but a interpretation that the activation of endogenous antioxidants would be mediated by a molecule or a factor of non-radical nature. So in 2015, we revisited the preparation for oxidative stress hypothesis and proposed that, in fact, these environmental situations associated with metabolic depression would, in fact, induce a overproduction of reactive species that would then activate the expression of antioxidants in animals that use metabolic depression as a survival strategy. In the following years, we then decided to take a look of how widespread is preparation for oxidative stress. Is it restricted to some animal group or a kind of environmental stress? And then we investigated the literature on 80 animal phyla and 6 different stresses associated with lack of oxygen. And what we found is that overall, preparation for oxidative stress is widespread in the animal kingdom, with at least one species in all 8 phyla that had been studied at that time. At least one species upregulate antioxidants in response to low oxygen stress. And here is the global view of 93 species that had been studied in these studies from 2016. More than 60% upregulate at least one antioxidant when exposed to lack of oxygen. So at this time we realized that all the studies, 99% of the studies were made with animals captured in the wild, brought to the lab, and then exposed to anoxia or hypoxia. So we proceed to answer the following question. Does preparation for oxidative stress occur under natural conditions? And this is what led us to the Brazilian semi-arid region to study frogs, estivating frogs. So we know from the literature, for example, in stress hormones, that animals in the lab behave and have biochemical response that are different from animals in the wild. So this was our main motivation. Is preparation for oxidative stress a real phenomenon? Does it happen in nature? Or it is something of lab animals? So we went to the Caatinga, which is a Brazilian biome. It is located in the northeast of Brazil. It has high temperatures and low humidity, but the main feature of the Caatinga is the unpredictability of the raining, of the intensity of the raining season. So here in this graph, we have in the x-axis the month, and in the y-axis the total precipitation or rainfall. We can clearly see a pattern that has a rainy season during the first half of the year, and then a dry season. But the unpredictability of the Caatinga comes from these atypical years when we have a much lower rainfall in some years. Some years we have a pattern that does not show the clear rain and dry season. So animals living in this region must survive this kind of unpredictability. And there are some regions of the Caatinga that animals stay for 3 years without a significant rainfall. So what some of the frogs in this region use is they enter in estivation. So here we have pictures of some frogs during the rainy season. They are happy, eating, reproducing. But during the dry season, the river streams completely dry out and animals bury themselves underground and depress their metabolism until hopefully the rainy season comes again. So we went to one of the driest places in Brazil, which is the city of Angicos. So we have here the northeast Brazil. Here's the Rio Grande do Norte state, and here's the city where we collected the animals in a place called San Miguel Farm. We went there by car from the capital of the state, and this is what we found there. We find 3 frog species: Pleurodema diplolistris, Physalemos albifrons, and Proceratophrys cristiceps. These three frog species bury themselves in the dry soil and estivate during the dry season. And what we know and we knew at the time of collection is that animals are not oxygen limited. This graph in the bottom left shows the depth of the soil and the concentration of oxygen. And we see that up to 1.5 meters, there is no hypoxia in the soil. And what we also know is that the longer the dry period in the bottom right, this graph, I apologize again, this is in Portuguese, but I'll translate it now. We have in the x-axis the time since the last rain, and in the y-axis the depth in which animals are found. So we have this tendency of finding animals deeper as the dry season advances. So animals are not exposed to environmental hypoxia, and they appear to move vertically as the dry season goes by. Here are some pictures of the collection site. During the rainy season, this is a river. I hope you guys can imagine, like, see, this is the river bed that is completely full of water during the rain season, and the stream goes this way. And during the dry season, the streams are completely gone. So there we first mark the surface to record the depth of capture of animals. And then we start digging with shovels, and hopefully we find some animals during this digging. We can find animals as deep as 1.80 meters, but sometimes we dig, and we find nothing. So here is an example of the trail left by one of the animals. Let's see. Laser pointer. Okay. So here we see here, and here is the trail left by estivating frogs. So as the dry season advances, animals butter deeper and deeper. So they are under metabolic depression, but they are able to move. And when we dig the soil here, this tunnel that was less compact, it just falls down. So what I mean is that animals do not have this column of air above them during estivation. This is just soil that was not as compact as the soil around it. Here just another picture. So a tunnel of less compact soil that estivating animals made. And during our expedition, as I said, there are 3 species that estivate there, but we focused on 2 species: Pleurodema diplodistris on the left and Proceratophrys cristiceps on the right. So these 2, although they survive the same environmental conditions, and we find them side by side in the same region. If we dig a hole in the dry riverbed, we find both of them. They have some interesting differences, and I have summarized it here. So Pleurodema is smaller than Proceratophrys. It moves faster, it consumes much more O2 both at rest and when it's forced to move. And more interestingly, Pleurodema rapidly awakes. When we are digging during the dry season, when we find one Pleurodema, it immediately open its eyes. As you can see here, this is a picture taken like a second after digging and finding one of them. It immediately open its eyes and it's apparently fully awake. So in the case of Proceratophis, this does not happen. Here is a frog that we dug out, we manipulated it by hand, put it in another surface, and it appears sleeping. So it's not as responsive as Pleurodema. And here's some characteristics of the collection site. During the dry season, the air temperature can reach 38 degrees Celsius. And more interestingly, the soil surface on the sand can reach temperatures above 50 degrees Celsius. But as we recorded temperature in different depths, we see that it is very stable around 32 to 33 degrees Celsius from 30 centimeters to 1 meter and 20 centimeters. And here, again, sorry for the Portuguese, on the bottom right, we have the depth in which animals were found, and the y-axis, the number of animals found. So we have a fairly normal distribution in the depth in which animals are found. So we went to this location on several occasions, but the results, the biochemical measurements I'm going to show you next, were made in animals collected in 2005, 2007, and 2012. So we have 4 sets of animals. The first set is a group of Pleurodema diplolister animals that were active. The second set of animals were estivating animals, Pleurodema diplolistris animals. Another set was Proceratophis cristiceps animals that were active. And the last group were animals that were estivating during the dry season. So we have 4 groups, 2 species, and 2 metabolic states at different years of collection. And more importantly, what we did, as I said, Pleurodema animals awake, they resume activity very rapidly. So our strategy to avoid an artifactual change, biochemical change, was to pick the animals and freeze in liquid nitrogen immediately. So this is what we did. We wanted to freeze the biochemical parameters as they were when animals were underground. So we brought animals back to Brasília, which is where our lab is based, and we dissected the skeletal muscle around the femur of these animals and measured several biochemical variables, which included metabolic enzymes, antioxidant enzymes, an oxidative stress marker to oxidative damage to proteins, and we also measure glutathione in its reduced and disulfide forms. So finally, to results. So what we found was that animals during estivation, it was already expected that they are in metabolic depression, and we already knew from previous studies that they consume less oxygen during estivation. And this was also reflected in the activity of citrate synthase, which is an oxidative enzyme. It's a marker for oxidative metabolism. And we saw a decrease in both species. So let me just go through this axis here. In the x-axis, we have active animals and estivating animals, active and estivating. And on the left, we have Pleurodema diplolistris, and on the right, Proceratops cristiceps. In green, Pleurodema is the animal that is more active, awakes rapidly, and consumes more oxygen when during the rainy season and during forced activity. So we confirm the depression of oxidative metabolism. This was also confirmed measuring the activity of isocitric dehydrogenase. And what I forgot to mention is that when we compare active animals from both species, the slower species has a lower activity of both citric synthase and isocitric dehydrogenase. We also measure the activity of PK as a marker of the glycolysis, and we didn't see much differences, just a slight difference between estivating and active Proceratops cristiceps. We then investigated anaerobic metabolism, even though we knew that underground there was no oxygen limitation. We wanted to check if the capacity for anaerobic metabolism was changed during estivation, and we saw no difference between estivating and active animals. Then we proceed to measure glutathione levels. On the left, I have total glutathione, which is the sum of reduced and disulfide glutathione. And for Pleurodema, we saw a tendency to increase, but it was not statistically significant. But in Proceratops cristiceps, we observed an increase of 57% in total glutathione levels. And this was very similar to what we observed for reduced glutathione, in which we had an increase of 61% only for the Proceratops cristiceps individuals. Here for the antioxidant enzymes, we had a huge variability in superoxide dismutase activity, regardless of the metabolic state or the species. There was a huge variation, and we didn't see any difference. But for catalase, we saw an increase during estivation for both species. We also observed an increase in GPX activity when using lipid hydroperoxide as substrate. And a very similar scenario was observed when we use hydrogen peroxide as the substrate. And for these three enzymes, catalase, total GPX, and GPX, the activity of what we would call a control active animal during the rainy season was lower in Proceratophis cristiceps than in the other species, Sclerodema diplodistris. We also measured the activity of glutathione transferase, and we did not see any change when comparing estivating versus active animals. But there was a huge difference of more than 10-fold difference between species. There's a axis break here. I hope you can see it. And there is this huge difference in the activity of GST. The degree of protein oxidation was very similar between these two frog species and was not affected by the estivation period. For the levels of disulfide glutathione, we observed a 65% decrease only for Sclerodema diplodistris, but this decrease was not significant for Proceratophis. And here again, Proceratophis cristiceps has lower levels than Sclerodema diplodistris. And lastly, when we calculate the percent of glutathione that was in its disulfide form, its oxidized form, we calculated the GSSG to TGSH ratio. We saw that estivating animals had a more reduced state in their skeletal muscle during estivation. And here again, the Proceratophis cristiceps has a lower level than Sclerodema diplodistris. So next, we took all this data and tried to reduce the dimensionality and made a principal component analysis. And we could clearly see that the experimental groups grouped together based on these biochemical variables. We have here the first component that clearly separated Sclerodema diplodistris on the right and Proceratophis cristiceps on the left. And the second component clearly separated active animals on the top and estivating animals on the bottom. And when we check which measurement, which biochemical variable contributed more to each of these components, we see that for the first component, which separated species, had a strong contribution of citrate synthase, isocitric dehydrogenase, and glutathione transferase. And for the second component, which separated active and estivating animals, we had a contribution of the markers related to redox metabolism. So we have a strong contribution for glutathione peroxidase and the GSSG to GSH ratio. So here is just to summarize, we compared animals from the rainy season, active animals, with estivating animals found underground. We focused on the skeletal muscle, more specifically the muscle around the femur, which was the region that that yielded more material. And what we found comparing species during only the rainy season, comparing control animals, we found that Sclerodema diplodistris has higher citrate synthase activity, isocitric dehydrogenase, antioxidant enzymes, and a more oxidized state. So this is the species that's more active, consumes more oxygen, and awakes very rapidly when disturbed, or when it starts to rain, it's the first species that arise on the surface. And when we compare active versus estivating animals, putting together both species, we saw an increase in glutathione levels, glutathione peroxidase activity, and catalase activity, which were associated with lower metabolism at the enzymatic level, a lower level of GSSG, and a lower degree of glutathione oxidation. Thus, going back to the question of whether preparation for oxidative stress happens under natural conditions, we confirmed this prediction. And that's it. I'm open for questions and discussions. I'll be happy to discuss with you.
Okay, Daniel, that was a very nice talk. But I know every little detail for work, so I will just open for questions, and maybe you can talk. Maybe I'll ask you a question, but not related to this work. But what are your plans after this? I know that you have a lot of plans, but it will be nice to share with everyone.
Okay, so currently, I kind of changed my experimental model to insects, and also came back to the lab and not in the natural conditions. And I am currently designing some experiments to elucidate the mechanism that we proposed in 2015. My aim is to use an insect as a model to confirm or reject our explanation gave in 2015 to explain the molecular events that lead to preparation for oxidative stress. Those are my plans, and we have a lot of samples in the freezer from these frogs too, so we have a lot of possibilities.
Okay, thank you. So please ask questions.
Marcelo, can you reduce the screen so to...
I'm trying to. Okay.
Okay, okay. Daniel, really, congratulations. It's very interesting works, no only one. And I like mostly that you are doing experiments in the field. Really, that's for me, that's mean adaptation. I have some questions about your work. The first one is I have seen, did you present some results indicating increased activity of salt during estivation in some species?
Yeah, in some species, yes.
Okay. How do you explain it? How you can explain ROS production during estivation?
Yeah, this is something we kind of avoided in our previous publications, because the biochemical conditions during hypoxia are one, and during estivation are other. So I personally don't have a good explanation for the case of estivation for those animals, such as these frogs, that we know do not experience hypoxia during estivation.
Okay.
There are some other frog species, such as the Australian Cyclohana, where the soil is muddy and they do experience some hypoxia. But I do not have an explanation for the biochemical mechanisms in the case of estivation.
It happens in many species, and Marcelo, probably you know very well that several investigations have shown that ROS are produced even during hypometabolism. And that's really a question. And I don't know whether there are several other systems, okay, which are involved in the ROS production. Did you determine the gene expression of SOD, for example, for SOD catalase, etc., before frogs enter estivation?
Yeah, no, we didn't make any gene expression experiments. But I believe that this kind of deeper investigation is more suitable for lab experiments, because we can control better the conditions and make several replicates.
Yeah.
Thus, we
It's a very
focus
nice
on,
indicator that really POS start early when I have a gene expression of antioxidant enzymes. And probably later some mechanism activate the translational phenomena. I don't know.
I have a proposal.
Yeah, okay, go ahead.
Yeah, it's just a hypothesis. There's nothing to prove. But I see these cases in estivation where there's no hypoxia. Yeah, I think it's a similar mechanism, but because to increase activities of antioxidants, you have to have a signal of increased free radical formation. And I will bet like $1,000 that will have also an increase in heat shock proteins in Danielle's frogs. But we didn't measure, but
Yeah.
it's for sure. And so, but what is going on? They are in hypometabolism, you know. So in these conditions, it is possible that this slower flux of electrons in the mitochondria may increase some leak of electrons. So in the beginning of estivation, it's possible that a little surge of free radicals may activate antioxidants and other response. It's a kind of hormesis, but no one has done that. There is just one work that I know that has been done. It is not published. It is a PhD thesis done in Singapore. Yeah, with lungfish. I think it's 2016. I have by Professor Ip in lungfish. So in the first days of estivation, it's all done in the lab. In this thesis, they see an increase, a little increase in carbonyls. I don't remember the tissue, but that is a kind of suggestion that there is a little oxidative stress, a physiological oxidative stress that trigger the increase in antioxidants, because in lungfish, there is published, very well established, that long-term estivation, you have an increase in antioxidants. But the process of going into estivation has not been shown. And they have this PhD thesis, but it was never published, unfortunately. I have it if someone wants to read. I can email. It's like 400 pages.
I know. That's I'm asking because they are very interested in data, so that's why I'm asking. And I don't know whether did you determine that the activities of the oxidant enzymes after arousal. Did you determine a further increase in the activities?
Yeah, we didn't have this set of animals at the arousal. We only have, because there is some work to be done to collect these animals. We are very geographically away from the collection
I
site.
know.
We have a very limited sampling points. So
Yeah.
we only have animals during activity in the raining season and underground during the dry season. But I believe it would be very interesting to to see what's going on at the moment of arousal during the first rains, or even artificially awake some animals that are during the dry
Okay.
season and offer water.
Okay. And this muscle type is mostly anaerobic.
No, it's
It's
aerobic.
aerobic.
Yeah.
You mean that there are plenty mitochondria in the muscles?
Yeah.
Okay. And another data I saw, and I like it, is that there is not changes in the PK and the LDH activity.
Yeah,
How do you explain it? It's did you can we say that we have a stable glycolytic potential or not?
yeah, I explained it as animals because first, just one comment. We are measuring maximum activity of the enzymes. So this is the capacity of the system to. We do not know the actual reaction rates in vivo, but my interpretation would be that the animals maintain this capacity for glycolysis and anaerobic metabolism to be able to respond to any immediate changes in the environment, so they can awake rapidly if needed, given that this region has unpredictable rainfall. This is how I would interpret this data. But for sure, we need more experiments and more measurements, because only with maximum activity of the enzymes, it's kind of hard to.
Well, that's a good explanation, but I think that there is one more explanation why these animals maintain a stable glycolytic potential during estivation. Is that because oxidized always glycogen, and glycogen liberates more water than fats, and that is an explanation. Okay, and they need more water during estivation. So this biochemical adaptation helps, and is involved in the water balance, metabolic adaptation. That's another explanation. Could be. I don't
It's
know.
a good one.
Yeah. That's Marcelo for me. Daniel, congratulations.
Thank you.
Thank you very much. Regarding awakening, there is a work from Professor Debas. He is attending the seminar from India. He did a very nice work. We can invite him. We're inviting him for a seminar in the near future about his toads. So he did controls, hibernation, arousal, short time arousal, and long time arousal. It was very nice work. we can talk to him and ask him to give us a seminar with his work. So we will learn a lot about this frogs from cold mountains in India, cold mountains. So it'll be nice. So we see what happens in arousal, long time arousal. Okay, everyone, thank you for your questions, Basile. Please, people, ask questions to Daniel. He's there for help us to understand his work.
Hi there, it's Costas. How are you? Hello to everyone. A really, really interesting talk. Daniel, congratulations. While you were showing us all these results, I was like saying, okay, this is a really, really a good work with lots of amounts of data. How do you think you should do you have something to do complementary to the tissues you have still in the fridge to, I don't know, to explain some things to yourself first and to complement the existing results? Because I was seeing your results and I was saying, okay, how many things we could do on this, you can do on these tissues that you have in the deep freezer. Which is the best way you think you can compensate, you can add to your thesis, to your research? I don't know what comes to your mind.
Yeah, our original idea was to so first we have all the tissues from all these animals in the -80 in the ultra freezer, and we have only consumed the muscle from one limb. So we have liver, we have heart, we have all the other tissues, and we still have muscle. So our original idea was to take a look at some transcription factors using Western blot, but our problem is that we would have to like we are not sure that the commercially available antibodies would work. Probably some for amphibians could work well, but my intention at that time, we even bought some material to do this analysis, was to take a look at the transcription factors at the protein level, which is also based to what equipment we had access at the time and now. So that's to where I would go. But today I discussed with Professor Marcelo some days ago that it would be interesting to take a look at the proteins involved in repair, in oxidative damage repair, to see how they are modulated in these animals, and maybe include some chaperones and heat shock proteins.
Okay, so that's
But
oh, sorry, Marcelo,
no,
I think Daniel meant DNA repair. You missed the word DNA.
yeah, but I think something like protein damage repairs, such as methionine reductase. Oh, methionine sulfoxide reductase. It's also a repair protein that I think would be interesting, not only DNA.
And this will supplement greatly everything you have done. But can I, from my experience, even I don't know if it's helpful for you, because we have worked with Vasilis Michailidis he was my professor, and he's my academic dad, as I call him. We have worked a lot on fish, a lot on bivalves, and also frogs. And we have seen that its antibody that we used and for fish worked also on frogs, so they have this consistency. I don't know if it's helpful
Nice.
for you.
Yeah,
Yeah,
it is.
but
It
we
is.
have, we have all the antibodies that we have ordered work greatly also on amphibians.
That's great news. Thanks.
Yeah. So thank you for your presentation. It was really, really amazing. I hope everything goes for you as you are planning in the future, and I hope the best. I wish the best for you.
Thank you very much.
And we'll talk in these meetings again.
Yeah, sure.
Thank you, Konstantin, for your comments. Thank you very much. So open for other questions, please, people. Do you want to complement anything with your experience in hibernating toads? Is your microphone working?
Yeah. Okay, thank you, Marcelo. I like that Daniel really show us differences between two species. Okay, if I understood it well. And really, that's a question. Is because of different biology, lifestyle of the species, different adaptational mechanisms for these two species. How you can relate these two species with the biology of the frogs?
Yeah. What we don't know very well this species. What I can tell is that they are very similar. For example, what they eat and the microenvironment they seek. And as I said, when we dig the soil, we find them side by side. But we do know that they have some physiological difference. As I said, one species consumes more oxygen than the other, and one species awakes very rapidly, and that's what we know. And this species, which is the Pleurodema diplolistris, which not run, but leaps faster, consumes more oxygen. It has a higher activity of metabolic enzymes and has higher activity of antioxidant enzymes, and it also has a more oxidized GSSG to GSH ratio. So that's what we know. We don't know much about the life history of this species.
That's really very important because you show us that citrate synthase, for example, in one species is almost a double activity than the other species.
Yeah.
Okay, really, it's a very good, the most important indicator of mitochondrial metabolism and aerobic capacity. So you can say that aerobic capacity, the second or the first species is higher or lower. Okay. And what about, for example, that's another point for me, about the other tissues, and especially the heart, which is full of mitochondria. Can we see, have you similar data or not? I don't know whether there are similar differences.
Yeah, we have the heart in the freezer, but we did not make any measurements so far, so we don't know. But we have this species to be
That's-
analyzed at some time.
I think that could be nice to complement your data with data from other tissues. Because
Yeah,
of
sure, I
related.
agree.
Okay. And of course, you didn't determine a lactate level in the tissue, in the muscle, during estivation.
I have
Even in the blood.
I did measure lactate, but it was a sample that was frozen and frozen several times. It was kind of a pilot experiment, but the results were that it did not change lactate levels in the skeletal muscle. But I chose not to present it and not even to publish because of this potential artifactual from freeze and thaw cycles. But what I saw was that lactate levels did not change.
Not science. Okay. So you can say that the biggest difference is in the aerobic capacity of the two species, between the two species.
Yeah, there is also one big difference that is related to glutathione transferase activity, which I also can explain. One species has 10-fold more GST activity than the other.
And the last question, can you say that is there any difference in the time of estivation between the two species? That's mean that the first one, for example, is more tolerant to hypoxia than the other one. You can say that.
No, we don't have this kind of information. What we do know is that the Pleurodema diploistris it awakes very rapidly. So even what the locals, the population, the communities that live in this area say is that the Pleurodema, which is the smaller one and the faster, the one with higher aerobic capacity, it awakes before the actual first rain of the season. It senses that the rain is coming. So that's the information we have, but we don't have information who enters estivation first, or how long does it take. We don't have this.
Okay. That's all, Marcelo.
I have something to add. You know, even though these species they dig in the sand, you know, they can be up to 2 meters. We did find at 2 meters in depth, but you can find lower. Yeah, the sand, it's the size of the sand in this place is big, so the air goes normally well up to 2 meters. Our friends, they measured the oxygen levels of 2 meters of depth, and they found this like tiny differences in of oxygen levels compared to the top layer, like 1% less. So they have plenty of oxygen. However, there is another group of frogs from another area. I mean nearby, like 300 kilometers. For Brazil, it's close. and these frogs, they dig in mud, you know, and they should be in hypoxia and deep hypoxia. It's not other species. We never had the chance to investigate these species there, but they should be very interesting. There's some fish in Brazil that also they estivate when the rivers they dry up some months of the year, and they probably in environments that are very mud. So probably they are in hypoxia. So these are interesting models for estivation plus hypoxia. But these two frogs, they're both no normal oxygen. They just depress the metabolism with plenty of air. It's just the option. They decide to depress metabolism. So it is different from the hypoxia model. It's just metabolic depression. Yeah. And I think we didn't do a very reliable lactate determination. But for our data, we don't expect to see an anaerobic metabolism increased in estivation, for at least for these species, at least in these conditions. However, it is possible that these very species may be hypoxic tolerant for another environment, the same species, but that we don't know. We did all the time, all the work in that farm, in that area. It's a research station. People they do real physiology. We don't do real physiology. We just get dead animals, dead frozen animals. But
okay.
Yeah.
But,
So
Daniel, really, you determined lower oxygen consumption during activation, which means that you have activation of an anaerobic component of metabolism. Okay. Under these conditions. It's okay that?
Yeah, no, we know that the whole organism oxygen consumption is reduced during activation, but they also lower their demands, energetic demands. So
Yeah. Okay.
we can't say that they are in anaerobic conditions, because we expect that the need for ATP is also reduced, so it's balanced. And we did not observe increase in lactate dehydrogenase, which would be for muscle. In vertebrates, we would expect that this would compensate for any need that was not supplied from oxidative metabolism.
Okay.
I didn't mention, but I also measure, since we were working with skeletal muscle, I also measure creatine kinase, and would be a source of ATP, and it also does not change during activation. So we didn't see any indication that there was a mismatch between supply and demand of energetic resource.
Okay. It is very interesting. I think it isn't the temperature or the failure of food, but only because you have a low rain time. That's the most important environmental factor that makes these two species to enter hypometabolism. Is that?
Yeah.
Thanks a lot.
Thank you.
Thank you, Professor Basile. Thank you. So just was one question. There's nothing to do with this. How long did you stay in Ken's lab? You know, for everyone, Basile and I, we were postdocs in Ken's story, and he's getting close to 80 years old. So I think it's in 3 years we should prepare some surprise, some like special issue about his work. How long did you stay there? I stayed 2.5 years.
Even.
How long did you stay there?
I stayed 9 months.
Oh, 9 months. Okay.
9 months.
Yeah, he was there before me.
Yeah.
He couldn't go. He couldn't attend today. He is teaching today. Mondays for Ken, it's not good. But he asked Daniel to send the slides. Okay, this discussion about oxygenation in metabolic depression is very interesting. I had some collaboration with Kelly Drew from Alaska. We did some work with squirrels in a hibernation, but just by pure biochemistry, redox biochemistry. But I follow some of these discussions they have about what happened to the oxygen in the tissues of squirrels when they hibernate, the Arctic squirrels, because they decrease metabolism by 90% or even more. And in the beginning, like in the 90s, they used to think that squirrels would be in hypoxia during hibernation, and they developed all these comparisons between squirrel hibernation and ischemia in humans, you know. But the years went by, and actually the data was the opposite. During hypoxia, looks like oxygen is higher than normal. Because, you know, the temperature is low, so it actually increases the solubility of oxygen, and the consumption of oxygen by the other tissues are decreased. So it looks like there is plenty of oxygen during hibernation. So people had to rethink everything. Everything has to be turned around. So it's an incredible area, you know, this change in the way we interpret or the way we hypothesize things, you know, when you face the real results, and we have to, okay, we were all wrong. We have to think something different now. But it is a fascinating area, fascinating area. And thank you for all your comments, Professor Basile. So do you still have time for some questions? For us here in Brazil, it's lunchtime. Do we have more questions? Do we have more questions? So if we don't have more questions, I will we'll thank Daniel for his presentation. So can everybody open the camera so we can take a picture of us? Okay. We'll do a print screen. Yeah. Hello, Alexandra. Hi. Oh, Professor Debas. Hello. Hi there. So we invite you. You know your microphone is not working. So we are inviting you for a seminar to present your data on your toads, your hibernating toads, in maybe December, whenever you like. Okay, it'll be a pleasure.
Okay. Thank you, sir.
Yeah. So
Thank
let's
you.
take a picture of everybody here. Okay. Thank you for everyone. So see you in our next seminar.
See you soon. Thank you. Thank you. Bye.
Bye.
Bye-bye.
Bye. Bye.